right. Okay. Well, welcome everyone. Thank you so much for coming to this breakout session. I know it's been a long day, and I hope to make this as enjoyable and as informative as possible. I'll leave plenty of time for questions at the end. And so we're just going to get started. I should start by at least introducing myself. My name is Donna Chang. I am currently the CEO of Hope Biosciences. We are a stem cell banking as well as a stem cell therapeutics research company based out of Sugar Land, Texas. And we've developed some very cool technologies that are currently in use in many, many different clinical trials for many applications. But I come to you today, first and foremost, as a mom. I am a mom to twins, 5-year-old twins, Sam and Grace. And somebody who is very passionate about the healthcare system that we're currently in and actively involved in kind of solving the problems that we see in medicine today and trying to be an active contributor in fixing some of those issues. And somebody who has studied cell biology. I have a master's degree from Johns Hopkins in this area, and I am currently actively involved in lots of research related to these specific cells. So with that, I'm going to begin with talking about stem cells. So we're going to take a biology 101 class really quick, okay, so that you all feel comfortable about the subject. It's important because when you talk to your physicians, it's necessary for you to also know these terms because a lot of times the doctors don't know either. Okay, so what are stem cells? Stem cells are cells that basically have no identity. So picture your baby. Your baby might become one day a police officer or a lawyer or a doctor. So they're full of potential. They can grow into anything. A stem cell is the same thing. It is a cell that has no identity. At first, something in the body needs to signal that cell to become bone, skin, hair, all the cells that make up your body. So an embryonic stem cell comes from an embryo. You guys have heard of embryonic stem cells? Okay, embryonic stem cells come from an embryo. And it's been long thought that these cells are going to be the cells that we're all going to use because they are capable of becoming an entire human being. But there are ethical and safety implications with using embryonic stem cells. And the beauty is that after we're born, we still have stem cells circulating in our bodies. How do you know that? Let's take an example of a cut. Let's say you get a cut. What happens? You bleed, and then you get a scab that forms, and suddenly a scar, and then eventually goes away over time. What you're witnessing is actually the regenerative potential of your own body. So through the blood, we have stem cells that are in circulation. Our stem cells are made in our bone marrow. And then those stem cells are circulating in the blood. And so when you get that cut, the blood delivers the stem cells to that area, and magically, those stem cells become new skin and just heals just like that. Now you notice over time, the older we get, the pace at which we heal slows down. That means that over time, we're losing the cells that are inside of our bodies. So the main question is, okay, and this, you're probably wondering, what does this have to do with baby banking? We're going to get there. At the end of the day, we want to be able to somehow find these stem cells that repair our bodies to fix ourselves one day. Isn't that the goal, right? We all want these magical stem cells. And what are they, by the way? This is our tagline. This is very important. Not all stem cells are created equal. Stem cells are all different. You have to ask the right question. Okay, what's the question we're asking? What stem cell is responsible for maintaining our bodies, for regenerating our bodies? It's not about the source. So when you hear about source, you'll talk about embryonic stem cells versus placenta. Oh, I want the placenta. I want the cord. I want the cord blood. These guys are all sources. They're not cells. You have to ask what stem cells are the ones that are responsible for regenerating. Okay, so now you're going to sound really smart because you're going to learn a new word, which is number 9 on your scavenger hunt, mesenchymal stem cells. Mesenchymal stem cells are cells in our body that basically maintain everything. It also manages our immune system. Immune system is very important for health. Okay, so your cells for autoimmune disease as well. We think that the future trajectory of pretty much anything chronic degenerative, any injury, any autoimmune disease, any vascular disease will be used, mesenchymal stem cells will be used in those cases. These cells are so versatile. Again, the problem is, as we age, we lose them. So, what if we could bank them? What if there's a way to store these amazing cells so that we could use them when we need them? So now the question becomes, how do you store to use? Okay. Because storing for the sake of storing is not fun. It's imagine you go to a bank and you store your money there and you never access it. That's not fun for anybody, and you shouldn't pay to do that. So you want to bank something that you know is going to be usable. So how do we do that? Well, first, where do you find these magical mesenchymal stem cells? Cord blood contains a different kind of stem cell. It's called a hematopoietic stem cell. So for those of you looking at cord blood banking, you should just know what you're banking. Okay, what you're banking are hematopoietic stem cells. What do those do? Those make blood. Do MSCs make blood? Do mesenchymal stem cells make blood? No. That's the one thing they can't make, but they can make everything else. Okay, so hematopoietic stem cells make blood. Therefore, they should be able to be used for blood-based disorders, right? Sickle cell anemia, acute leukemia, things like this. Currently, there are approvals to use cord blood in those instances. However, if the child, you've banked cord blood, and that child ends up growing up to have a blood-based disorder, they cannot use their own cord blood. They have to use a donor, because in that sense, their cord blood is not making healthy cells, so they have to get a donor cell. Does that make sense? Okay. Mesenchymal stem cells, again, if you want to be able to use your own, you have to be able to localize them in some source. So in adults, we can find it readily available anywhere, but we actually take it from fat tissue because everyone seems to be really thrilled to give it to us. That's why we select fat. From the baby, again, we're trying to figure out the most non-invasive, easiest way to be able to attain these cells. That's the placenta. Now, why not the cord tissue? You've heard of banks. You'll hear about cord blood as well as cord tissue. They're absolutely right. Cord tissue and the placenta both are good sources for mesenchymal stem cells. That is true. But we find that the placenta amniotic membrane is the easiest place to isolate these cells consistently. You get very beautiful MSCs from that. But if I were to take those cells and stick them in a freezer, there's no guarantee. There's no way to know whether they'll be viable in 20 years. So let's say that child grows up, they're 80. Let's say they grow up to 80. Okay, they're one day going to be 80 years old, and they have early onset, they have some kind of Alzheimer's disease, and they need to access their cells. How are we going to know? How are we sure that those cells are going to survive and be able to be useful? Well, bottom line is, if you take these cells at birth and you stick them in a freezer, you're not going to know. It's impossible to know. So the only way to solve this problem is to do the hard work at the beginning. That means at the time of birth. So now you've given birth. This arrives in this beautiful kit, comes to our lab. In order to make these cells, we then have to, from the placenta, isolate the MSCs. We have to find them. Okay, it's about 100,000 again. You see this thing at the tip? That's basically the amount that we start with. And then from there, the cells are microscopic. And we're going to seed it on a flask. And this is a culture flask. This is a rather large culture flask. The culture flask we use at the very beginning is a little itty-bitty one. And on that flask, the cells grow on the bottom of this flask. Okay, they basically take a small piece of real estate and then they just expand throughout the entire flask. When we start growing these cells, and again, we have to grow them because they're not enough to make a therapy of any kind. The amount of cells we'll isolate from the initial tissue is about 100,000 cells, which literally would fill the tip of this cap. It takes 12 of these, full of these, to make one treatment. Okay. So in our clinical trials currently for chronic degenerative diseases, we do a lot of work in neurology. You need 200 million cells. That is the dose that we have designated with FDA as a therapeutic dose. It takes that many to make a therapeutic difference. So the cells, once they're seeded, will grow, and then we will cryopreserve them. We'll take them in and we'll create what's called a master cell bank at every generation that we're growing these cells in. And it takes 2 months. So that legwork, that process of growing cells takes 2 months. Once we finish that, and we've successfully done that, we can send you a certificate with your actual cells, your baby stem cells. That's what they look like. And they are safe and stored. And then those cells can be used in the future. Anytime cells are needed, our patented process allows us to take a vial of the cells, seed a new flask, and then repeat the growing process. That's how you get repeatable treatment. How many treatments? We can make 1,037 treatments from that one piece of amniotic membrane from the placenta. So that, we think, is enough cells for the rest of your life. That's why we say it's lifetime banking. We are looking out for the course of the entire child's lifetime. Today, we look at disease. We're studying disease. We are, pretty much focused on fixing problems. But I think by the time, for my Sam and Grace that are currently 5 years old, I think that maybe by the time they're 25, 30, their access to these cells are not going to be just for treatment of a disease. I think the way we have to look at this is these cells can be used to hopefully prevent disease, and that's exactly where we're trying to take this. So that's our long-term goal. We were not a storage company to start with. We're a therapeutics development company, but we do recognize that really the first part of making a treatment starts with good cells. You need good, healthy cells that are banked in order for them to be usable in the future. So that's what we do. And it's important to know the difference. So always ask the right question. What kind of cells are these? How do you know they're viable? It's one thing to know that they're viable. Okay, what does viable mean? Viable means alive. Okay, but there's a lot of degrees of being alive. You can be alive and functional, and you can be alive and non-functional. There's lots of different definitions of that. So when it comes to making a medicine, you have to be able to look at the activity of the cell's not only biological activity, but their ability to do all of the functions that we know they can do, like regulating the immune system and being able to differentiate and being able to do all the things that they are destined to do. So with that, I think I will start answering some questions because I know there will be a lot. We're going to use it to find the stem cell. We are pure stem cell banking. We do not bank tissues or blood. Okay. Largely because we have not seen that it amounts to enough cells to make a therapeutic quantity that'll work in the future. Even
the cells themselves, they're never really frozen.
Our cells?
Yeah.
Once we create enough of them, right, we're going to spend 2 months making these cells. Once we create enough of them that we feel okay with, then we're going to go ahead and freeze those cells because we're already anticipating that when we thaw them, when that child needs their cells and we thaw it, a lot will be dead. But that's okay because there's going to be enough alive to make new treatment. That's exactly right. That's exactly how you think about it. Yeah. So at the time of birth, there are instances where that can happen, or let's say the hospital will also keep the placenta. It's possible that happens. If they do, then there's no way we can do it. If that placenta is tested, it may stay at the hospital for 3 or 4 days and then released. We can give it a shot. No one pays for anything until we know we successfully banked. That's our peace of mind guarantee. We're not going to bank. We're not going to just try this and then not let you know. You will know. You will get the certificate and you will know that it's done. Rest assured, it's done. And we think delayed cord clamping is very important. Okay, why? Cord blood contains hematopoietic stem cells, right? So you want those hematopoietic stem cells to feed back into babies. It's probably the reason why so many babies are born anemic. So they shouldn't be. We should make sure all that blood, take the time to make sure that blood goes to the baby. Yeah, no effect at all. Placenta. Yes, sir.
In the case of
Not for banking. Banking is really it's a commercial activity because it's your choice to bank your own tissue. Later on, when these treatments get approved, they're going to be approved in 2 settings. Either you getting to use your own cells or you using a donor cell. Okay? Donor cell meaning some other healthy person that's donated tissue or placenta or whatever to make the cells. That instance, eventually insurance if it's FDA approved, insurance will cover. Today, right now, there are no stem cell treatments, like stem cells that have been manufactured that have been approved in the United States. None. Okay. Cord blood, again, in its natural form, is available for use only for certain blood conditions. Okay. Yes. So cord blood, again, contains hematopoietic stem cells, so just blood-based diseases, okay? And if you look at mesenchymal stem cells, they can do the rest. So there's a pretty...
I found this on the web.
Oh, Siri has an opinion about that. But yes, I think that as right now in current regulation, because what we do here is actually drug production. So that's why everything we have to do has to get cleared by the FDA before we administer to a patient. Okay, and we've already released about 2,000 treatments in this last 3 years. So when we look at the application of these cells, I think it's only going to become more and more as the years go by, because it's, I think, not accurate to look at this as a particular treatment for a specific disease. Unfortunately, that's the way it is right now, but eventually it'll be sort of a tool, a regenerative tool that we use for everything. Yeah, so one day it'll be that way. All of the cells that have been used so far, all the babies that have been banked have not had to use them yet, okay? So we don't want them to use it. But those are adults. So from adults, we isolate the same cells from the placenta from the fat. So if you wanted to bank, we can take your fat. The babies that bank from their placenta cells will never have to give fat later. And so then it begs the question, well, if you can bank later, then why should you bank at birth? Well, because you never know what can happen. And that's sort of this whole biological insurance thing. But I just know that at this time, the cells that we isolate from here are superior to the ones we find in fat. Okay, they are. So far from what we have personally measured in our lab, there is more cytokine production, which means our mesenchymal stem cells excrete. They're like a drug factory. They make tens of thousands, from what we know, compounds they excrete. Okay, all kinds of stuff. Lots of anti-inflammatory cytokines. They're basically like a drug factory. So the cells from a placenta, the mesenchymal stem cells from a placenta, are the same as the ones from fat. However, these ones express more. So there's more growth, there's more proteins and more cytokines that are excreted from a placenta than there is from fat. So slightly better. Will that make a therapeutic difference? I don't know. Until you do a clinical trial, the answer is no one knows. But I think it's a unique opportunity. And then they don't ever have to worry about it in the future. I think like it's a whole nother generation of kids right now that are going to grow up with this option. So, yeah. So mesenchymal stem cells, these MSCs, are immunomodulatory, meaning they're regulating your immune system. So if my cells went into your body, they would play nice. They won't create like a reaction. Now, scientifically, that's sort of known across the board. And in our clinical trials, we have not observed any kind of reaction. Now, I myself personally believe that though science thus far tells us it's safe to use a donor source, and I think for acute conditions, let's say you have an accident and you have a spinal cord injury, yeah, we should immediately treat you. There's no question about it with a donor cell if you're not banked. That's absolutely the way we should go. But long term, right? One day, if our kids are going to be using their cells once a year just to maintain themselves for the rest of their lives, then using their own is the best, because there's never any potential for rejection. I just feel like we're at a point where, 100 injections later, could they be rejected? We don't know. It's never been done. Yeah. So for spinal cord injury, to give you an order of magnitude, like right now we have 5 trials in spinal cord injury, and it takes about 40 infusions to get people to have sensation back in their legs and to have actual meaningful clinical differences. So that's the order of magnitude we're talking about, which is a lot of cells. Yeah, billions of cells. Yes. Yeah, if you go to hope.bio and you'll look at our clinical trials tab, you'll see all of the different uses our cells have been used in. Again, we started in neurology because that is a primary area of an unmet need. So Alzheimer's, Parkinson's, spinal cord injury, traumatic brain injury are all applications that we have used cells in. Most recently, the Department of Defense has authorized a $5 million grant for our second trial in traumatic brain injury. So that means that the risk of car accident is always there, right? And risk of head injury is always there. And so in order to be able to use cells for this sort of thing, you have to be able to prove that they work in these things. So we think that right now it's sort of we're at a cusp here. Like we're just at the starting point, and you'll see more and more application in these cells. But you really need a technology that can support that. You need cells in the right quantity. You always have to remember that. How many? We should be asking, how many cells are you banking? And how do you know that? How do you know they're viable? How do you know they're going to be useful? These are questions we should be asking, because that usability is going to translate. It definitely is. We're already there. And many people are now involved in trials and participating in trials using these cells. So we're just at the beginning, but you'll see it'll grow very soon. Adults can sign up as well. You can. Our website has lots of banking information for both adults and newborns. We bank primarily adults. Newborns are really sort of newborns actually from the placenta. We had tried and failed multiple times. You've heard other companies taking different sources like placenta and tissue and the cord tissue and freezing it. Again, we would try. We would get these frozen tissues and try, and we would find very little viable cells, enough to be able to grow. So we've been trying this for a while. My son and daughter are numbers 2 and 3, successful bankers. So that's how new it is. It's just within the last 5 years. Yeah. Right now, I think we are making good headway. I think what happens is you'll see a lot of similar kind of stem cell products fail along the way. And I can tell you the reason for failure always has to do, I think, with viability. Okay, meaning if the cells are not viable in every way, again, not just we measured it and they're alive, not just to that level, then you're not going to know whether they're going to be actually efficacious in a trial. If you ask your doctors, do stem cells work? They say, it's never going to amount to anything because it doesn't work. Most of the time in trials and large-scale trials, it doesn't work. But we are achieving, for the first time, we are achieving consistent results because we're not doing frozen products. Every time we make a treatment, we're going to take one of these that we banked at birth, we're going to thaw it out, and we're going to reculture. Because this is what we do. Culturing is our jam, okay? That's what we do the best. We are, all of our technology and what we've patented is our ability to make perfect stem cells. So we take these and we reculture them and we deliver them fresh to the doctor. So that's how treatment will look like in the future. I love the question of vitamins because it's like, how many blueberries do we have to eat to prevent cancer? It's like one of those age-old questions. You're told you should do A, B, and C to prevent disease, and how are we really going to do that? And so, what's amazing right now in terms of the day and time we live in, is that there's a lot of good technology pouring into not only you can do it. Before you do CT scans and MRIs, you do it to find a problem, right? But now, there are places that you can go and you can just go get a CT scan just to look at your overall health, right? And so with these tools, we can actually start to, for instance, if I take a 50-year-old, a healthy 50-year-old, and you look at their brain and it's functioning perfectly, can we test to see if stem cells can help optimize that brain, right? That's sort of the prevention we're looking at. How can we keep ourselves healthy and our tissues healthy in order to sort of survive the aging process? And the aging process is a degenerative process over time. So that's what we're looking at. And those are measurable. Those are quantifiable. So we'll be looking at trials like that in the future. Yeah.